So I extend my greetings to each and every one of you who is joining today's call. Welcome. Thank you for joining this presentation of the HIV Prevention Trials Network 083 primary study results. We are so delighted to have you with us and grateful for this opportunity and the responsibility that we have to disseminate study results to the public, to our collaborators, stakeholders, partners, and others. So I am Melissa Turner. I'm based here in Washington, D.C. I'm co-chairperson of the Community Working Group of the HPTN. Our network's mission is to reduce HIV incidence worldwide, and we endeavor to conduct high-quality ethical research that is supported, guided, and informed by community. So today, we gather for the presentation of primary results of HPTN 083, which is a global randomized, controlled, double-blinded study that compared the safety and efficacy of long-acting injectable cabotegravir to daily Truvada for pre-exposure prophylaxis in cisgender men and transgender women who have sex with men. And just a few months ago, we met to share the preliminary or interim analysis and the evidence that was available at that time that both study products in 083 were effective. Protocol leaders for 083 today will present primary findings and final outcomes on today's webinar. And these findings have been described in the media already as impressive, appealing new option, revolutionary, groundbreaking, a game changer, and we all agree. We believe in the power of an ethically sound, community-guided, and well-designed, controlled clinical trial to advance prevention science. That's our fundamental premise here in the HPTN. And the 083 results presented today is certainly an important step forward towards that goal. So I just want to extend my gratitude and recognition to all of the many hands that have facilitated 083 to this point in time, where we're able to present these primary study results today. And on behalf of the network, I just want to extend gratitude to all of you who have made this possible. We want to thank our community partners, our collaborators, stakeholders, local, national, and globally. We thank you all for the guidance and consultation and partnership that you provided as we've carried out this study. And certainly want to thank and lift up the 4,570 who enrolled in 083. These volunteers are just remarkable. They accepted the terms of the study. They provided consent. They invested their personal time. They tolerated the risk. They gave of themselves very courageously and generously. And we thank them for the contributions that they've made. And this is a study cohort that is very well represented by Black men who have sex with men, transgender women, and young people under 30. It's a very impressive and diverse study cohort. These participants were very engaged with clinical research sites, staff, and as demonstrated by the high quality data collection and the strong retention for a study of this size and length. It's very exciting for our network to be able to present this evidence for a new tool in HIV prevention today. And so on today, we will hear from the scientists leading 083, and we will follow their presentations with questions and answers and discussion. We're using the chat feature solely given the large number of participants anticipated on the call. We do have leadership from our companion study for 083, which is 084, evaluating cabotegravir amongst women in sub-Saharan Africa. This is an ongoing study in the field, and we've included leadership from 084, given that it is a companion study and very important activity of our network right now. And so I'm pleased to introduce our moderator for today's webinar. He's a treasured supporter and collaborator for the HPTN, Mitchell Warren. For 16 years, Mitchell Warren has been the executive director of AVAC, Global Advocacy for HIV Prevention, which focuses on HIV prevention research advocacy. communications and stakeholder engagement. And Mitchell is a favored moderator of HPTN webinars. We thank you again for being with us again today, Mitchell, and I turn the webinar over to you. Thank you.
Great, Melissa, thank you so very much. And to the community working group, which is core to the HPTN. And I'm delighted, although we'll hear a lot of science today, as an advocate, I'm delighted that you are driving this and invited me, and just grateful for the partnership. Because in the midst of great science, in the midst of thousands of trial participants, it's advocates and communities that need to move this research forward and act on research results. And so just thank you so much, and delighted to be with everybody. We have a lot of people on the line, which is fabulous. I am going to do my best to bring as many of your voices into the call as possible. And as Melissa said, we are going to rely on the Q&A and chat feature in Zoom, given the large number of people. Please do feel free to start those questions and conversations as you hear presentations. I'm going to, in just a minute, introduce the leadership of HPTN 083 to present in succession data that were presented last week at the AIDS conference, but now we have a chance to hear it live and in person, and then to spend the latter half of the call in a conversation with them and with other colleagues, as Melissa described, and I'll introduce them in a minute. So again, I'll try to my best to put those into the queue, and as we've done before, want to commit with the HPTN, AVAC, and others to be sure that whatever questions we don't get to, we will do our utmost to answer those offline after the call. So without further ado, I'm delighted to welcome both Rafi Landovitz and Beatrice Grinstein to the webinar to present. They have been the leaders of this trial from its inception. I think many of you know already Rafi as the protocol chair based in Los Angeles, California, and wears many hats, but for today's purposes is protocol chair. And Beatrice is calling in from Rio de Janeiro and is a leading researcher based in Brazil, but a leading global researcher, and is the protocol co-chair. Rafi is going to present the primary results of the study in the first presentation. We'll go straight to Beatrice after to talk about what the results look like at a geographic level with regional and various key population breakdowns. Again, we're going to go through both of those presentations, and then we'll get into discussion, but do keep your questions coming as we go. And without further ado, I'm delighted to say good morning, early morning to you, Rafi, in Los Angeles, and take it away.
Thank you. Thanks, Mitchell, for that very kind introduction, and good morning, or good afternoon, or good evening, depending on where you are, to everyone. I also want to thank the CWG and Melissa for that really kind introduction, and to thank all of you for joining us today. As both Melissa and Mitchell said, without community engagement at every stage of this study, we certainly would not be where we are and moving this field forward in such an exciting way. So thank you for the partnership, as both Melissa and Mitchell have already said, and we all echo. Can everyone see the slides okay? Is the screen sharing working?
It is.
Okay, great. Because many of you know that I'm technologically challenged. But regardless, this is going to be a recapitulation of the presentation that we did at the AIDS 2020 meeting last week. So for those of you who heard it once already, I apologize for the repetition. And for those of you who didn't get a chance to hear it, I look forward to sharing this information with you on behalf of Dr. Grinstein, my co-chair, and the rest of our HPTN 083 study team. These are the final results of HPTN 083 pre-exposure prophylaxis containing long-acting injectable cabotegravir is safe and highly effective for cisgender men and transgender women who have sex with men. These are the authors of the abstract, and these are my financial disclosures that it's important to keep in mind in understanding this presentation. What I'm going to go through today is I'm going to briefly provide some background and context for the origin of the HPTN 083 study. I'm going to review the HPTN 083 study design, even though many of you are pretty familiar with it already, and its objectives. I'm going to summarize the statistical methods And then I'm going to present the HPTN-083 study population findings about HIV incidence overall and by each study arm, safety of both study products, the details of HIV acquisition events, some additional outcomes of interest, including sexually transmitted infection rates and weight changes that we saw in the study, and a number of critical analyses are still in progress. And I'm going to go over what we're still working on and waiting for, and give you my sense of why these are really important to a full understanding of these study results. I'll then finish with some summative comments and some observations. So it's now really well known, and certainly by everyone on this webinar, that tenofovir-based preparations in combination with emtricitabine are highly effective products for HIV prevention across populations and contexts, with a series of rigorously conducted randomized clinical trials demonstrating high levels of protection against HIV infection when they're used as prescribed, and a margin of forgiveness to non-adherence against rectal protection, but much more rigorous adherence, of course, required to maintain vaginal protection. Despite these clinical trial successes, the public health benefits of pre-exposure prophylaxis scale-up haven't been universally realized, with young adults, Black and African American and Latinx, MSM, and transgender individuals continuing to not only be disproportionately affected by HIV, but also by challenges with PrEP uptake, adherence, and persistence using current PrEP regimens. Recent advances in pre-exposure prophylaxis have included the development of a monthly dapivirine ring with efficacy against vaginal acquisition, which is currently under regulatory review in Europe, and a decision is expected imminently, and the approval of daily oral TAF-based PrEP by the US FDA for gay men in this past October. HPTN-083 was aimed to build on accumulating evidence for antiviral activity of long-acting injectable cabotegravir, which is a novel integrase strand transfer inhibitor. And this efficacy has been clearly demonstrated when used in combination with long-acting injectable rilpivirine for maintenance of virologic suppression in persons living with HIV. This combination of long-acting agents is currently under regulatory review in the United States and is approved in Canada. We compared a PrEP regimen that contained long-acting injectable cabotegravir to daily oral TDF-FTC for HIV prevention in a phase 2b/3 randomized double-blind, double-dummy clinical trial that was conducted at 43 sites in North and South America, Asia, and Africa. The study enrolled cisgender men and transgender women who have sex with men at ages 18 and above at elevated risk for HIV infected, as defined on this slide, who were generally in good health, had no evidence of viral hepatitis infection, no history of seizures or seizure disorder, and had no contraindications to gluteal intramuscular injections. The planned enrollment was for 5,000 participants, and pre-specified key population metrics, as noted by Melissa in her introduction, included that at least 50% of participants would be under the age of 30, at least 10% of participants would be transgender women, and that at minimum 50% of the US enrollment would self-identify as Black or African American. Further details of the study outcomes in these key populations will be presented by Dr. Grinstein as soon as I'm finished with these primary results presentation. The study's primary efficacy and safety endpoints are also listed at the bottom of this slide. The HPTN-083 was a study with 3 distinct phases or steps. After one-to-one randomization to either cabotegravir or TDF-FTC, participants began step 1, where either an active cabotegravir tablet and a TDF-FTC placebo tablet were administered, or an active TDF-FTC with a placebo cabotegravir tablet were administered. This was to be taken daily for approximately 1 month to establish safety and tolerability of the short-acting cabotegravir formulation before any administration of the long-acting formulation of the drug. Participants with at least 50% adherence to daily oral tablets by pill count and acceptable safety profiles advanced to step 2. And step 2 was the blinded comparison of oral and injectable study products. Cabotegravir arm participants received a single 3 milliliter gluteal intramuscular injection of cabotegravir at a dose of 600 milligrams that began at week 5, again at week 9, and every 8 weeks after that. And they continued their daily oral TDF-FTC placebo tablets. The participants who were randomized to the TDF-FTC arm continued to receive active, although blinded, daily oral TDF-FTC and received placebo injections consisting of a 20% intralipid feeding solution, which was visually indistinguishable from the active cabotegravir product. And the injections were given on the same schedule as the active injections in the cabotegravir arm participants. This oral injectable comparison continued for 3 years after enrollment. All participants who discontinued injections prematurely were provided open-label TDF-FTC for up to 1 year after their final injection to provide ongoing HIV protection during the tail of long-acting cabotegravir. Participants were then linked to local HIV prevention services. All participants had frequent safety laboratory assessments, every 6-month asymptomatic sexually transmitted infection testing. They were offered testing more frequently if they were symptomatic, adherence support, risk reduction counseling, including condom and lubricant provision, were at all study visits. Going forward, I will use the color convention that's on this slide of cabotegravir arm results being shown in orange and TDF-FTC arm results being shown in blue. HPTN 083 was designed as a non-inferiority study using a relative non-inferiority margin of 1.23, an alternative hypothesis for the cabotegravir versus TDF-FTC hazard ratio of 0.75, and aimed to enroll a population with a background HIV incidence of approximately 4.5 per 100 person years. The sample size was predicated on a population whose adherence by detectable plasma tenofovir would be approximately 57%. That's actually a typo on this slide. The study was planned for 172 incident events to be fully powered with pre-specified interim analyses when 25%, 50%, and 75% of endpoints have been observed with extremely rigorous criteria for early stopping. The study was reviewed at regular intervals since its inception in December 2016 by an independent Data Safety Monitoring Board, or DSMB, at the NIH. The first interim analysis with 25% of endpoints having been accrued occurred on May 14th, 2020. At that DSMB review, the board recommended stopping the blinded portion of the study for having met rigorous criteria for definitive demonstration of high levels of efficacy for cabotegravir compared to TDF-FTC. They recommended public dissemination of the results and that participants be unblinded and offered cabotegravir as soon as such supply was available to do so at the sites. At the time of the blinded study stopping, 4,570 participants, as Melissa mentioned, had been enrolled and randomized. The intention to treat cohort excluded 4 participants who did not meet inclusion-exclusion criteria of the protocol, and therefore include 4,566 participants. The primary efficacy cohort was pre-specified to exclude participants, 5 participants in this case, who were found to have been HIV infected prior to exposure to any study product, and an additional 71 participants who had no follow-up visits at which HIV status could be determined. Retention overall was approximately 90% at 6 months in both arms, 86% at 12 months, 80% at 18 months, and 75% by 24 months. The intent to treat study population is described on this slide. Key populations were generally well balanced across randomized treatment arms. You can see specifically that 567 or 12.4% of the overall population was represented by transgender women. Remember, we had pre-specified that at least 10% would be transgender women, and fully 2/3 of the participants were under the age of 30. 50% had been pre-specified in the protocol, and there was a mean overall age of 26. Regional and enrollment distribution is represented also on this slide, and Dr. Grinstein will be going into more detail about this in her presentation. Approximately 50% of the US enrollment self-identified as Black or African American, as was pre-specified in the protocol, and the race and ethnicities of the other regions are also represented in this slide. Again, Dr. Grinstein will be going into more detail about this in her presentation. These are the primary study results. Overall, we observed 52 incident HIV infections in a total of 6,389 person years of follow-up. Individual participant median follow-up was 1.4 years. This resulted in a pooled HIV incidence of 0.81 per 100 person years. And in the lower left panel, you can see that in the cabotegravir arm, we observed 13 HIV incident infections in 3,202 person years of follow-up, yielding an incidence rate of 0.41 per 100 person years. In the TDF-FTC arm, we observed 39 incident HIV infections in a total of 3,187 person years of follow-up for an incidence rate of 1.22 per 100 person years. These incidence rates yielded a hazard ratio shown in the lower right panel on this slide of 0.34, and the 95% confidence interval on that hazard ratio is 0.18 to 0.62. And what I'd like you to particularly take note of in that lower right panel is that I've identified 3 critical values that are excluded by that 95% confidence interval. It excluded not only all the way to the right, the pre-specified non-inferiority margin of 1.23, the unity result, which defines superiority of 1.0, but also our pre-specified alternative hypothesis of 0.75, resulting in a definitive and unequivocal superiority result for this study. Showing here is the cumulative incidence plot of the intent to treat population. The plot demonstrates the timing of the infections in each arm, each step up in the curves representing 1 incident HIV infection in each study arm, and the highly statistically significant result of a hazard ratio of 0.34 with a p-value of 0.0005. The next series of images, although somewhat complicated, really bear careful attention. I'm going to go through it in a lot of detail. These describe the current state of our understanding of the 13 incident HIV infections identified in participants receiving cabotegravir, and also the 2 participants exposed to cabotegravir who were subsequently found to have been HIV infected before any study product administration. I want to really preface this discussion by noting that we do not at this time have the results of drug concentration testing or resistance testing for any of the incident or prevalent HIV infections in this study. That work is proceeding as rapidly as is possible during the current SARS-CoV or COVID-19 pandemic. And we look forward to presenting those results to you as soon as they're available in a peer-reviewed setting soon. To orient you on this slide, I have grouped the 15 cases into 4 categories that I found helpful for thinking about these cases. The proportion to the right of each case number indicates the pill count-based adherence recorded by the site during the step 1 oral lead-in. The numbers above each segment represent the weeks between the time points that are depicted. The little syringes represent the timing of research site administered injectable study product. The hatched orange lines represent a delay in injectable study product administration, and the hatched blue line indicates the approximate date that the participant would have been anticipated to run out of TDF-FTC had it been taken daily as prescribed. The gray segments indicate periods of time at which, again, the participant would not be estimated to be in possession of any study product because the participant had not attended the study visits at which they would have been dispensed. Daggers to the left of the study, the case number, represent HIV acquisition events whose dates are still not final due to ongoing back sample testing at the HPTN Laboratory Center. The first group I'm going to talk about are cases A1 and A2, and these represent the 2 participants who were found to have been infected with HIV before administration of any study products on sensitive back testing of stored samples. These incident HIV infections were not detected at the site until the time of study product discontinuation, where you see the lines end in these 2 case plots. In the case of participant A2, This delay in diagnosis at the site level resulted in administration of one active CAB-LA injection in the context of already having acquired HIV. Cases B1 to B5 represent participants who were found to have HIV infection after a prolonged hiatus from their last cabotegravir administration. Two of these participants, B1 and B4, had been provided open-label TDF-FTC after premature discontinuation of their injections. B1 was lost to follow-up and then represented over 1 year after the last TDF-FTC supply had been prescribed and was found at that point to have acquired HIV infection. B4 was in the middle of a 1-year course of post-injection TDF-FTC when they were discovered to have acquired HIV infection. Cases C1 to C3 represent HIV infections that occurred during the oral lead-in period. Two of these were delayed in detection at the site and therefore were also exposed to injectable cabotegravir after infection had been established but not detected at the site. Pill counts of all three participants do not by themselves explain these HIV acquisitions. Detailed HIV detection assays and molecular clocking studies are being pursued to establish as precisely as possible the exact timing of the acquisition and exposure that led to these infections to clarify the timing of onset of protection of oral cabotegravir. Cases D1 to D5 are of particular interest because as far as we are able to establish at this time, they acquired HIV infection despite excellent adherence to oral cabotegravir, and with the exception of one 10.7-week interval between participant D1's 5th and 6th injections, they received their injectable cabotegravir as scheduled. Detailed pharmacokinetic analyses of these participants will be critical to the understanding of what circumstances were permissive to HIV acquisition in these cases, as they will be with all of these incident cases. Some of you might note that without CAB drug concentrations and resistance profiles, these cases are extremely difficult to interpret, and we look forward to sharing those results in a peer-reviewed setting, as I mentioned, as rapidly as possible upon their availability to have a full understanding of how to contextualize these results both for providers and potential consumers of this product. As is known to be the case with TDF-FTC PrEP, delays in the reactivity of HIV diagnostics, including nucleic acid testing, can complicate identification of HIV infection in the context of ARV-based PrEP use. It appears that CAB is no exception to that observation. One final modification with animation on this slide allows you to see with the asterisks the timing of the first detection of the HIV infection at the site level, which was delayed substantially compared to when it was detected with extremely sensitive and multiple assays performed at the HPTN Laboratory Center in 7 of these 15 participants. There are these are the 39 incident and the 3 prevalent HIV infections detected in participants provided TDF-FTC. Again, we do not yet have the results of gut drug concentration testing nor resistance testing for any of these participants. Given the observation of 3 early infections occurring in the oral cabotegravir arm during that oral lead-in phase, it's noteworthy on this slide that the earliest infection detected in the TDF-FTC arm occurred 10 weeks after entry and provision of TDF-FTC oral study products. Given the high levels of protection that TDF-FTC, when taken as prescribed, is known to afford, we assessed adherence to TDF-FTC in HPTN 083 to establish a benchmark by which to interpret the TDF-FTC arm incidence rate of 1.22%. Detectable plasma tenofovir concentrations were found in 87% of samples from a random adherence sample of approximately 400 TDF-FTC arm participants for whom data from 372 are currently available and shown on this slide. 75% of these samples had plasma tenofovir levels over 40 nanograms per milliliter, which correlated with high levels of protection in randomized controlled trials. Additionally, we assessed intraerythrocytic tenofovir diphosphate from DBS samples in this adherent subset and found that about 75% of samples overall contained concentrations suggestive of 4 or more doses per week of TDF-FTC on average over the 1 to 2 months prior to the assays being conducted. Interestingly, in the adherent subset across the green bar at the top of the left-hand side of this figure, you can see that the proportion of participants over time who had tenofovir diphosphate concentrations consistent with high levels of rectal protection did not vary tremendously, staying in the 60 to 80% range over at least 81 weeks, in notable contrast to clinical cohort and open label data. I will now turn to adverse events. Significantly more participants in the cabotegravir arm experienced injection site reactions, nearly 81% of those who received at least one injection, as you can see in the overall numbers on the left side of this slide. I'm being told that my internet connection is unstable, so if people are having trouble hearing me, please someone interrupt me and let me know, and I will try and call back in using another mechanism. But for the moment, I'm going to try to keep going unless someone tells me that they're having trouble hearing. Also shown on these slides that the majority of the injection site reactions were mild to moderate and appeared to decrease over time with repeated injections. Small proportions of the injection site reactions were grade 3 or severe, and 47 participants, 2.2% of CAB participants who received at least one injection, permanently discontinued injectable study product due to an injection-related adverse event. The severity of the injection-related adverse event was strongly associated with the odds of permanent study product discontinuation. The injection site reactions were 60% pain and 23% tenderness, and the most commonly began a median of 1 day after the injection was administered and lasted for a median of 3 days. The primary safety outcome is presented here. Grade 2 or higher adverse events were experienced by 92% of participants overall, not different by treatment arm. Statistically significant adverse event categories experienced by 5% or more of participants in any study arm are shown here. These included an increased frequency of decreased creatinine clearance in the TDF-FTC treated participants and higher proportions of participants in the cabotegravir arm with nasopharyngitis, increased fasting and non-fasting blood glucose, and pyrexia or fever. The majority of the fever events occurred within 7 days of cabotegravir injection administration. Grade 3 and higher adverse events occurring at a threshold of 2% in any study arm are shown here and are not different between study arms. Serious and expedited reporting required adverse events, which included seizure and suspected drug-induced liver injury in HPTN 083, were rare and not different between study arms. There were 11 deaths in the study. All were assessed by the site investigators as not related to study product, except for one cardiovascular event death who had been assigned to the TDF-FTC arm. Prevalent bacterial sexually transmitted infections were frequent in the HPTN 083 study population, and incident sexually transmitted infections are shown here were not different between study groups. The incidence rate of syphilis centrally adjudicated is 16.5 per 100 person years, and aggregate rectal STIs were over 27 per 100 person years, indicating that participants were at ongoing risk for HIV acquisition via condomless sex acts throughout the study conduct period. We had previously reported, and I would orient you to the right side of this slide, that in the HPTN 077 study, which was a randomized placebo-controlled phase 2 study in cabotegravir in low-risk men and women globally, that approximately 1.5 kilograms of weight gain was seen annualized from the cabotegravir treated participants. That was not different from the weight gain observed in placebo-treated participants based on observations from a 1-year time period. In HPTN 083 overall, we observed an annualized 1.3 kilogram weight increase from baseline in the cabotegravir treated participants, and a statistically lower 0.31 kilogram weight increase from baseline in the TDF FTC treated participants. What's really interesting about these findings, however, is if we break apart the first approximately 1 year or 40 weeks of the HPTN 083 conduct from later time points, we find a highly statistically significant difference in the first 40 weeks that then goes away. Over the first 40 weeks, very similar to what we saw in HPTN 077, which I still have up on the right side of the slide, we found about a 1.5 kilogram annualized increase in weight from baseline in CAB participants and a 0.5 kilogram annualized weight loss from baseline in the TDF FTC treated participants. Again, highly statistically significant. But from week 40 to 105, we found a remarkably similar, approximately 1 kilogram annualized weight gain in both arms, suggesting that there's a 1 year period of weight loss caused by the TDF FTC, but after that, a parallel and similar increase in weight for both arms, both groups in HPTN 083 after that 1 year time point. Work that is ongoing and is currently in progress includes this really critical data of drug concentrations of each PrEP agent and assessment of resistance profiles for all HIV isolates for all participants who acquired HIV in the study. These are all in process and of course crucial to the understanding of how best to achieve virologic suppression in the context of rare HIV acquisitions despite the use of cabotegravir. Our conclusions are listed on this slide. Both cabotegravir and TDF FTC were highly effective for HIV prevention. And I cannot emphasize enough that although we see here that this is a clear superiority result with a 66% reduction in the risk of HIV infection observed in participants receiving CAB to those receiving TDF FTC, that I do not want people to impugn the efficacy of TDF FTC. TDF FTC as a PrEP agent works extremely well if taken as prescribed. We do, however, have 66% fewer HIV infections in the CAB treated participants despite a highly effective TDF FTC product. I think that's extremely important as a take-home message here. Cabotegravir was extremely well tolerated, as both agents were, even despite injection site reactions. Happily, these were often mild to moderate and infrequently led to discontinuation of the injectable study product. We really need these peri-infection drug concentrations and a detailed analysis of resistance profiles to fully understand and contextualize these results. Cabotegravir is the first long-acting injectable agent to demonstrate robust HIV prevention efficacy in cisgender men and transgender women who have sex with men. We eagerly anticipate the results of our sister study that's ongoing, HPTN 084, comparing cabotegravir to TDF FTC for cisgender women in sub-Saharan Africa. It's currently ongoing, as you all know, with an incredible team led by Sinead Delaney-Moretlaway and Mina Hosseinipour, as well as adolescent bridging studies that are currently in the field or in development. Before I end, it goes without saying that to conduct a massive study such as HPTN 083 not only takes a village, it takes a metropolis. And I want to take my final moments of speaking to you to recognize our incredible study sponsor, the Division of AIDS at NIH and its funding institutes, ViiV Healthcare, who provided additional support, the HIV Prevention Trials Network, our pharmaceutical partners at ViiV and Gilead, and specifically our colleagues at DAIDS, the leadership of the HPTN, the Network Leadership and Operations Center, including Andrea Jennings and Kyla Gomez Feliciano, and in particular, our lead clinical trials manager, Mary Beth McCauley, without whom none of this would be possible. And you all know and love her. She deserves enormous credit, not only for this, but for putting up with me as a protocol chair. Our remarkable and incredible community program management team, Cheryl Blanchette, Jonathan Lucas, Marcus Bolton, our communications and finance teams, the HPTN Lab Center and the HPTN Statistics and Data Management Center, our incredible study team, our site community educators, recruiters, and CAB members, our site investigators at each of our 43 sites, our valued colleagues at ViiV and Gilead, the HPTN Black Caucus, in particular, Christopher Hawks Ortiz, our colleagues at ViiV and Gilead, and of course, the truly dedicated and meticulous teams at each site, and the site staffs who conducted an incredibly complicated trial with impressive fidelity, and of course, our study participants and their families. Although I'm extremely sad that we weren't able to take live questions during the AIDS 2020 participation, I look forward to answering your questions and engaging in dialogue with you all here. If you have questions that we don't answer, you of course can email me or tweet me after we are finished. Thanks so much for your attention.
Fantastic. Rafi, thank you so very much. Extra credit points for getting the Twitter handle Doc in a Box, which has always been impressive to me. Lots of great questions coming in. Please keep them coming. And we're going to get to them as quickly as we can. But first, delighted to turn it over to the HPTN 083 protocol co-chair, Beatrice Grinstein, who's going to take us through a little bit more of the data related to geography and population. Beatrice, are you there?
I'm here trying
Great.
to share my screen. Just a second.
Okay. While you're doing that, I know some questions just on logistics. I believe these slides will be made available online, and the HPTN can confirm that. And there's a really important comment from Julie Franks about how to make these materials come alive for CABs and communities and others outside of the scientific world. And I think that's a great point. And I know the PTN is on that, and I will just put a shameless plug. We at AVAC just put out yesterday kind of a primer, an advocate's guide to these results. I'm working very closely with the HPTN 083 team to get it right. So, but it's a really important point about the kinds of information flow.
So good morning, good afternoon, or good evening to all of you, depending on where you are. On behalf of the HPTN 083 chair, Rafi Landovitz, and Mary Beth McCauley, the lead clinical trials manager, we want to thank very much Melissa and Mitchell for your kind introductory words. I also want to thank very much to Melissa and the entire community working group and Mike Mitchell Warren and the AVAC for organizing this webinar. And many thanks to all of you for joining us today. Go ahead, please. So I also want to thank all co-authors listed here. And these are my disclosures. Okay, go ahead. So despite major advances in treatment and prevention, the HIV pandemic continues to pose a major global public health challenge. The HIV epidemic disproportionately affects key populations in the different countries and locations. These populations are usually underrepresented in HIV prevention trials. In previous PrEP studies, challenges with adherence to daily oral tablets have been observed in these populations. Not coincidentally, these are the same populations most affected by HIV. Next. So just a very brief overview, as Rafi explained it very well. HPTN 083 is an ongoing phase 2b/3 randomized multicenter double-blind, double-dummy clinical trial evaluating the safety and efficacy of a PrEP regimen containing long-acting injectable cabotegravir compared to daily oral TDF-FTC to prevent the sexual acquisition of HIV in cisgender men who have sex with men and transgender women who are at increased risk of HIV infection. In HPTN 083, study participants were randomized in a 1-to-1 ratio to one of the two study arms. The other arm provided active Truvada and placebos for cabotegravir. Cabotegravir was administered daily by mouth for 5 weeks and then via intramuscular injection at 8 weeks intervals after an initial 4-week interval load. Participants in both arms were then offered open-label daily oral Truvada for 48 weeks after their final injections. Enrollment was pre-specified to include at least 50% participants under age 30, 10% transgender women, and 50% Black in the United States. The blinded study The blinded portion of the trial was discontinued at a planned interim DSMB review on May 14, 2020. The primary results of this analysis have been just presented by Rafi. Well, next one. In this analysis, we aimed to describe the pre-specified key subpopulations enrolled in HPTN-083 and the HIV incidence and efficacy of CAB-LA versus TDF-FTC in these subpopulations. Next. A total of 4,570 HIV uninfected participants were enrolled between December 19, 2016 and March 16, 2020 at 43 sites in Africa, Asia, and North and South America. 4,566 were included in this analysis. Next one. No, go back one. Overall, the median age was 26. Almost 70% of participants were younger than 30 years old, and 12.4% were transgender women. Of note, 50% of the participants enrolled in the US were Black African Americans. Next. In Latin America, with sites in Argentina, Brazil, and Peru, enrolled the largest number of participants by region. Next one. Of note, the median age of participants from South Africa was 23, with 75% of participants younger than 30 years of age, and 10% transgender women. Next one. In Asia, sites in Thailand and Vietnam also enrolled a young cohort of participants and contributed with the largest total number of transgender women enrolled in the study. Next one. As previously noted, 50% of enrollment in the US was Black African Americans. Next one. No, go back. As you can see in this slide, retention at 12 months in the US was similar between Black African Americans and non-Black. Next. Again, it's very important to highlight that 70% enrolled participants were younger than 30 years of age. Of note, the burden of rectal STIs was higher among the younger participants. Retention at 12 months was higher among those older than 30 years of age. Next one. Overall, transgender women were younger and predominantly enrolled in Asian and Latin American countries. Next one. Retention at 12 months was similar regardless of gender identity or sexual orientation. The burden of rectal STIs among trans women was also quite high. Next one. As you can see in this plot, CAB provided high levels of protection among US Black, transgender, and young MSM globally, consistent with overall study results. Although no statistical inferences can be drawn, the directional and general magnitude of effect is preserved in all subpopulations. Overall, 52 incident HIV infections were observed, 85% of them among participants who were less than 30 years of age. Next one. In the adherence subset of approximately 400 randomly selected TDF-FTC participants, 70% had tenofovir diphosphate concentrations consistent with high level of rectal HIV protection. Lower proportions of participants with protective levels were observed among Black African Americans, transgender women, and among those participants enrolled in Latin America. Next one. A major accomplishment to be highlighted in HPTN-083 was the extraordinary community engagement and community educational work that led to the enrollment and retention of these highly vulnerable populations. And you can see in the following sites picture from some of the sites in the different regions showing some of the activities performed. You can go ahead and show the next slides, please. In conclusion, HPTN-083 is the first study demonstrating efficacy for an injectable long-acting PrEP agent. Pre-specified populations historically underrepresented in PrEP registrational trials were successfully enrolled, fostering inclusion and equity in scientific development. CAB provided high levels of protection among US Black globally, consistent with overall study results. Although the trends are in line with the overall results, no statistical inferences can be drawn. Lastly, I would like to take the opportunity to thank the community, the study participants and team members, our research sponsors and partners, and our DAIDS, NIH, VIV, and Gilead colleagues for their participation and support that led to the success of this study. You can go ahead one, please. From the initial study concept and to the execution of the trial, the commitment and dedication from these groups has been nothing short of exceptional. Through the inspirational efforts of our community engagement and educational teams and the Black Caucus, this landmark study was able to successfully enroll at-risk MSM and transgender women globally, our most underserved populations. Next one. Next one. Next one. And also, I would like to thank very much to all our study participants. Thank you very much.
Great. Beatrice and Rafi, both thank you so very much. Obviously, the clock ticks and we only theoretically have a few minutes left. I don't know about the HPTN. I'm certainly happy to keep this conversation going because it has yet to begin and lots of important questions. I know people are going to have to drop. This call is being recorded and the slides, as you saw in the chat, will be made available. Delighted to, in addition to having Beatrice and Rafi available for questions, Mina Hosseinipour, who is the protocol co-chair of HPTN 084 is on the line. Her colleague Sinead Delaney Moretle from South Africa had to jump off. In addition, we have Kim Smith and Alex Reinhardt, both from ViiV Healthcare, the developers of cabotegravir, and obviously already a lot of questions there. I do want to jump right in though, and maybe Rafi, turn it to you first. A lot of questions since May 18th, and certainly even just in the last hour, about our terminology. People, you know, it was a non-inferiority design, and that's what you all presented back in May. Now we've gone into superiority. There was a great question about the numbers you put up earlier, the difference between those, the 1.23 margin and the 0.75 margin. So if you could, in lay language, help us understand the terminology, superiority, non-inferiority, what the trial actually answered, and perhaps most importantly, how as a community we should be communicating what we know about these two PrEP agents.
Yeah, thanks, Mitchell. It's a great and really complicated question, and I want to thank you and others who have been working with us to figure out the best way to message this. So just so you know, the design of the study was non-inferiority, but superiority was pre-specified as a potential outcome in the study, and its metrics were defined as well before the study started. So when the study first was stopped by the DSMB, it was certainly crystal clear that that a non-inferiority boundary had been crossed. What we did not appreciate at that time is it actually was a superiority result in that moment. And the reason we didn't appreciate that, and we're very careful not to say that until we were 100% sure that that was the case, is that it's an extremely rare circumstance, A, that a trial stops at this extremely early time point with this degree of imbalance between the events in the arms. And it's also unusual to have a non-inferiority design, primary design study where the alternative hypothesis hazard ratio is 0.75. Those two events together made it unclear even to statistical experts at that moment that the result when the DSMB stopped it actually was a superiority result. And it took a lot of internal discussion to lay that plain and make sure everyone was agreed on it. And I do want to point out, just so anyone who is wondering if there could have been any commercial bias in the interpretation of those results, the NIH, SHARP, DAIDS, and HPTN teams discussed and came to this conclusion and vetted it with the DSMB before we shared any of that information with either pharmaceutical partner. So they found out, our ViiV and Gilead colleagues found out about our final interpretation of those data once we were confident in its results, and I want that to be clear. The superiority issue really needs to be interpreted in the context of the agents and the trial and the population. And I think, as I tried to enunciate in my presentation, we know TDF-FTC works extremely well when taken as prescribed. And we suspect, although I do not have today to share with you, that the reason for these 39 permissive infections in people randomized to TDF-FTC will be because they weren't taking it frequently or possibly at all. But again, we need the drug concentrations to support that hypothesis. I think we all were concerned that TDF-FTC being so highly protective would be an extremely high bar to meet or pass. And the fact that that cabotegravir did in this study, I think poses some really interesting and important questions that remain to be answered. Is there some intrinsic difference in the efficacy of the drug cabotegravir compared to the drug Truvada? You wouldn't think so. It's possible. We don't have all the data yet. You would hypothesize that what this study shows is that taking the act of having to remember to take a pill every day removes a barrier to some people realizing the highly protective benefits of PrEP agents. That is my hypothesis. We really need these drug concentrations to fully be able to validate that and have the full understanding. That's why I want us all to be very careful about that. I hope that's helpful, and I look forward to working with you all about figuring out how to message that to communities. My take home is both products work really well. If there is a context in which someone thinks that an injection might be preferable, or they think they might have challenges taking a pill as prescribed, then this might be a better option, because in this trial, it did perform in a superior way.
Super helpful, Rafi. Let me ask just on one of the last points you made about adherence, which we've always known matters hugely for every intervention. You presented both some data earlier in the presentation on the subset analysis of PrEP adherence, which looked very high. And I just want to be clear, and then maybe you're going to have to repeat yourself, but in the 39 infections in the active oral arm, you don't yet know anything about their drug levels. Is that correct? So although there was high adherence in the sub-study, we don't yet know about adherence and drug levels in those who got infected. Is that correct?
That's exactly right. Because the adherence subset was pre-specified, those drug concentrations were being run as we went along in the study. The people who acquired HIV, we had to wait until the study was unblinded to be able to begin that pharmacologic work. So we're a little bit behind in that. So you are 100% correct. That is a random sample of TDF-FTC participants. It may or may not include any of the people who acquired HIV and TDF-FTC. We will be having all of the drug concentrations for the TDF-FTC seroconverters, and that's in process.
Great. Some additional questions to come on the trial, but I do and I realize we are going to continue to go, so people please stay with us. I do want to bring in Mina to provide a quick update, because obviously, before I ask a question from the very beginning about what next for the product, obviously what next for HPTN 084, and questions about what you know and when we might be having a webinar with you presenting results. And I hope Mina can unmute.
Hi, thanks. I'm Mina Hosseinipour, the 084 co-chair. At this time, the HPTN 084 study is just at 95% enrolled. We're at 3,033 participants of our 3,200 target. At the DSMB meeting in May, we were advised to continue. There were no safety concerns at that time, but we had not accrued sufficient person time to evaluate the data fully. As many people know, we started a bit later than 084 or 083, and we also had a brief pause due to the dolutegravir issue and neural tube defects, which has now been found to not be a significant issue. So we are continuing in the COVID era to enroll and finish enrollment as to the best of our ability. We are, all of the sites are in operation right now, and we hope to have our next DSMB is planned for November, and we'll see what that shows. I think it's critical that we continue the study so that we have appropriate options for cisgender women.
Absolutely, Mina, thank you. And I know that everybody on this call and everyone involved in HIV prevention is rooting for your metropolis of 084 to answer the question as expeditiously as possible to complete this picture of the product. And maybe that's the perfect segue to bring in. I know both Kim Smith and Alex Reinhardt from ViiV are on the line. Obviously, and you will know before this week, before last week's results and before this call, but certainly on this call, people wondering what next for the product itself, and trying to understand the licensure pathway in terms of how you take these results, what happens concurrently with 084 as it continues until we have the fuller data set. So I don't know if Kim or Alex, you're able to come off mute and provide a sense of timing and what we can all expect.
Hi, Mitchell, it's Kimberly. Can you hear me?
Yes, I can. Thank you. Welcome.
Thank you. So I'm Kimberly Smith, Head of Research and Development for ViiV Healthcare. Nice to hear everybody's voice, and thanks to Rafi and Beatrice for those great presentations. It really summed everything up. One, I just wanted to make a couple of quick points and then address some of the next steps. So from the beginning, there was a commitment in our partnership with DAIDS to do the two studies, 083 and 084, in order to answer the question of the efficacy of cabotegravir in men who have sex with men, transgender women, and cisgender women. The studies were really designed around the same time. The delay in the start to 084 was due to some discussions with the regulatory agencies about alternative designs and some delays in regulatory approval in countries, outside of the US, where it took a little bit longer. And so it ended up starting about a year later. And then, as Mina mentioned, there was some interruption in enrollment in 084 because of some of the concerns around neural tube defects with dolutegravir and some additional protections that we built into the trial. And so obviously we're very excited about the data from 083 and want to, look to get, this product approved as soon as we possibly can. But we are committed, as has been said, I think a few times, we are committed to getting a broad indication that includes men and cis and transgender women. And so that is our desire, that is our goal. And so in order to do that, we have to work with the FDA and other regulatory agencies around the world to really see what that will take from their perspective. Obviously, the data from 084 is going to be a key component to that. And so, again, that just really requires us to sort of wait on some of the 084 data and negotiate with the regulatory agencies about what exactly that package of data needs to look like in order to allow that broad indication. I hope that answers the question. If not, please give me a follow-up.
Great. Well, Kimberly, that is helpful. I think many will have follow-ups about, is there a date and when can we expect it? I'm guessing the actual timing, as you say, is based on the next conversations you have with the regulators, correct?
Yeah, I mean, there's obviously a lot of variables with the timing. First, even just the timing of getting the data all together from the DAIDS group and from SHARP over to ViiV to allow the process to begin for submission. That will take many months, and then from many months to pull the submission together. And so in the meantime, obviously 084 is continuing to move along. And so I can't put any time frame on exactly when we would file. In general, we think the timing is probably somewhere around the first half of next year, but that would really depend on, again, those negotiations with the regulatory agencies and some of the data from 084.
Kimberly, thank you so much. That's really helpful. And I think, this is a moment for all of us as advocates, product developers, researchers to really use it as a learning moment about regulatory processes. We all decry in all science by press release. We certainly see too much of it in the COVID-19 era. But also, it's not as if regulators regulate on press release. And what you were just saying, the compilation of all the data, the study reports, it's not as if you've just shared with them Rafi's and Beatrice's great presentations and said, we have great data, approve. I think this is a chance for us
that
really
would be neat if we could do that. We should be advocate for that.
Exactly. We want full information. I think that is a helpful thing that we can help all of us kind of really learn what has to happen over the next many months as you file. And a question, I guess this is also, for you, Kimberly and Alex, as well as for Rafi, Beatrice, and others. And I know there's a lot of, in your last slide, Rafi, a lot of unanswered questions that you still need data on. But obviously, a question just came in about the oral lead-in. How will it be incorporated outside of clinical trials? And of course, I think, as you said last week, or maybe Sinead said last week on a video about this, no conversation of CAB-LA is complete without talking about the tail. And I think it is helpful for the hundreds of people in the line to hear maybe from both the 083 team what we do and don't know, and also maybe from ViiV what might and might not be possible with respect to both oral lead-in and the tail going forward.
So I can start with the question around the oral lead-in. As I think most folks on this call know, we are developing cabotegravir for treatment in combination with long-acting rilpivirine in addition to the PrEP indication. And so we are, again, working with the agency to really get to a point where the oral lead-in becomes optional as opposed to a mandatory requirement. And so, those same discussions will happen with the agency with regard to PrEP as well as treatment. So, again, ultimately in the long, I'm not sure how long it will take for that agreement to occur, but we've had some of these conversations with them around treatment. Again, we'll plan on having the same conversations around PrEP, and with the goal that we would ultimately have this oral lead-in be optional. And with regard to the tail, I probably, I think we can talk about
Is that the tail wagging the dog?
I said tail, and the dog said something about the tail must have been related to him. So the one thing that I will say about the tail is just that, the coverage of the tail as it's designed in the study is, is clear and was, exactly important to do as we were learning about the tail and learning about the effectiveness of cabotegravir. Ultimately, in the real world, the need for coverage of the tail is really quite debatable. If a person goes off cabotegravir and continues, for whatever reason, and continues to have risk, then there would be a need for them to be on, on some sort of PrEP. And so they're on PrEP, not so much for coverage of the tail, but to continue to prevent HIV acquisition. Mm. It's also possible a person would go off of cabotegravir because their risk profile changed and they were no longer at risk. Again, under that circumstance, there wouldn't be a need to, quote unquote, cover the tail. So I think it's a complicated discussion about, what that will look like. And again, how that will show up in the product label is part of the discussion that we'll have with the agencies.
Really helpful. I don't know, Rafi or Beatrice, do you want to add anything on either of those topics?
This is Rafi. No, I think we've obviously been in discussions with ViiV about these exact issues, and of course defer to them on discussions with the agency about labeling. And we look forward to being able to provide ViiV with the data broken down by the oral lead-in and the injectable phase in terms of whether we're able to derive any information about the extent of essentially risk mitigation that oral lead-in does provide, and whether or not it's necessary, and combine that with what we're able to learn from the onset of protection in the oral phase, and then give ViiV all the tools they need to go to the FDA with that information and a discussion about it. The tail, I think, is something that we all, I think, worry about a lot. And as Kim said, if someone remains at risk, they need to be on multiple prevention options that will continue to work for them if the injectable isn't one of them. And understanding the clinical consequences of the tail, that is, if in fact, what the resistance implications of HIV is acquired during the tail, and what drug-drug interactions are important, and when they stop being important, and of course the issues around pregnancy and breastfeeding, all are things that we need additional information to fully inform. So it's a really interesting and important question, and we don't have all the data to support an exact recommendation for how we think this should best move forward at this time.
Great. I know we are, and we're going to try to wind down. A couple of quick questions I'll try to throw out, and then I'll close it up and hand it back to Melissa. But there was a question early on in your presentation, and and a lot of, I think, high degree of congratulations to the 083 team for the diversity of the trial population, which was by design, I know, in the protocol, in terms of targets for age and race, and inclusion of transgender women. And someone asked a question about why, quote unquote, targeting 50% in African Americans. But I wonder if maybe Rafi or Beatrice could describe just really briefly how you got to that, those criteria and those targets, and why they're there.
Yeah, no, thanks, Mitchell. I'm happy to start, and then Beatrice can make sure that I got it right. This was a really critical discussion for us, because obviously I think we've all been acutely aware of the disparities in HIV acquisition rates, particularly in comparison to who is currently uptaking daily oral TDF, FTC PrEP, at least by prescription rates. We wanted to make sure that these results would be generalizable to the populations disproportionately affected by HIV globally. We had extensive consultation with community. We had great input from our community program managers who led us in this effort. We had great consultations with the HPTN Black Caucus. We took advice from the DAIDS Transgender Working Group. And it was really these extensive community and advocacy consultations that led us to saying that really we wanted this broad community engagement and partnership, and these would be the minimum acceptable contributions of enrollment to this study that would allow these results to be maximally generalizable. And that's why we set them, and we're really, really proud of the collaborations with all of the sites and our community partners that we were able to, we hope, set a new standard for representation by most at-risk populations in these sorts of prevention efficacy studies.
Super helpful. I think it sets a new standard, and I know that as advocates, many of us calling on this being the new normal as a requirement. So really grateful for that and impressive. I know we're not going to probably have time, and I don't know, we hopefully have some trial participants on the line, but it may be something we can do together going forward. I think a number of questions and comments came in about hearing from participant experiences. And I think both on the diversity point of view as well as on product experience. We now have some very real world examples, and I think giving voice to that, certainly in the social science that the PTN has done, but looking maybe to have a webinar, I need to ask about that, and maybe even understanding of results as both 083 and 084 participants. So clearly a lot of conversations we need to create going forward. I'm just looking, I know a lot of additional questions came in, and I'm going to hand it back to Melissa in just a second. There was an issue, and I think it relates a bit to the adherence one, Rafi, but the issue of, in contraceptive land, we talk a lot about perfect use versus, actual use. And a question came in about, do we know what is the true efficacy, perfect use of either of these products coming out of this study? And wonder if you might help explain a bit of that terminology and that experience.
Yeah, unfortunately, we don't have that information, right? What you would need to be able to have that information is you'd need a placebo arm or a companion cohort where you knew the true background HIV incidence in that population, and then you could, have essentially a true efficacy result, or well, you'd have still an effectiveness result because you'd have an incidence as used. And then you could try and restrict your analyses to estimate what a true efficacy result would be with perfect use. Because we don't have a placebo arm in the study, we're even yet one step further removed from that. We're working on a number of what are called counterfactual estimates of what that might be, but those are obviously difficult analyses to do and always have biases and limitations. So we look forward to finessing them and getting them as solid as they can be, and hopefully sharing them in a peer-reviewed setting upcoming. And when you're trying to figure out how the two drugs compare to each other with quote-unquote optimal use, it's also a challenging exercise to do. You can only really attempt it once you have the drug concentrations available, which we don't yet, but you might anticipate that they would both work extremely well. And there are going to be pros and cons to either one that I think is going to be very powerful in offering people choices in the future. That's what I hope is the outcome of this work. And of course, I hope for a similar result in 084.
Great. We're close to the top of the hour. Obviously, this is just one of many conversations that we need to organize over the days, weeks, months to come. There's a lot to do, as you say. I want to maybe just end on that last issue of choice and maybe provide a couple of quick summary thoughts, and I'll hand it back to Melissa. And again, want to thank, and apologies, we haven't had a ton of time for conversation, but want to thank the leadership of 083, the leadership of 084, the leadership of the HPTN overall, and ViiV for being on this call, and obviously we need to have many more conversations. I maybe just to quickly summarize some of the things that have been said or thought on this call. Adherence matters. It mattered a lot before CAB-LA. It's going to matter a lot with. I saw a tweet earlier today from a colleague who said, oh, now with this result, adherence won't matter. And I think you saw in the data that were presented here, as well as from commentary here, going to the clinic every 2 months is an adherence issue. It's different than daily pill taking, but adherence will matter for every product, and there's a lot we still need to know about both of these products, oral PrEP and injectable, and adherence issues. Superiority is a statistical term. It's written into the protocol. It's part of the scientific discourse that doesn't reflect real people's lives. Right now, CAB-LA outside of a clinical trial isn't even possible. It's not an approved product for anybody. And oral PrEP is, by definition, it is an effective product today. We hope that we will get to those additional products. I think what we can say, and I think Rafi, you described it so well, that we now have a third product coming out of efficacy trials in antiretroviral-based prevention. Actually, 4 officially: oral PrEP with TDF-FTC for all populations at risk, available today. Oral TAF FTC approved for people who aren't at risk of vaginal exposure, and a new trial being designed. We have the dapivirine ring sitting with a regulator now, and now we have the first of 2 efficacy results of CAB-LA. Each of these products is different. We really are in a better place than ever. And maybe just to pick up on something, Kimberly, you said about the regulatory pathway, the access planning has to begin now, as we all want to make these options choices for people. So obviously a ton of work to do. As is said very often in the HIV world, much accomplished, but I'd say today much, much, much more to do together. And looking forward to that collaboration with all of you on the line, and really delighted to hand this back to Melissa and have you close us out, because the community working group's engagement with these trials and looking to the future is going to be critical. So thank you all. Keep questions coming, and let's make sure we keep the conversation coming.
Thank you, Mitchell Warren, for that stellar moderation, and to all our panelists and presenters today. We appreciate you being with us, and to all our attendees. This has been a very interesting conversation, and just a dynamic learning experience just in the chat here. And we will have to work towards more of a 90-minute, I think, timeline to accommodate more interaction, because clearly there are good questions here, good discussion to be had, and more to come. And again, we want to just remind everyone that as a network in the HPTN, our work is unfinished. I think I want to emphasize most here is that we want to support the work of our 084 team as they endeavor to reach the enrollment goals for that critically important study that is operating in tandem with a worldwide COVID pandemic right now. And those teams are working just incredible, courageous work, protecting participants, protecting the staff, and pivoting and altering some of their site activities and processes and procedures to accommodate the needs of protecting participants throughout a pandemic. And I just want to hold them up and emphasize that it is really important that we support that work going forward. And we celebrate today. We are very proud of the accomplishments of 084, but there's still more to do. And we are hungry to press forward. We do not want to take our foot off the gas. We want to press forward to the next scientific finding and breakthrough. For our goal is to reduce incidence worldwide, not just for one population, but for all who are vulnerable. And so in that spirit, we hope to see you all again in a future webinar for new results. So do stay tuned. Thank you and be safe.